My name is Sharon Minsuk, and I heard about this meeting because I saw the announcement and it said, item 4, bring stuff if you have it. And I guess they wanted all of us to, but I happened to email somebody back about that announcement, and they replied and encouraged me, be sure you follow item number 4. So, hence I brought something. And I'm going to try to do it fast. I haven't talked about this in a while, actually, and I didn't prepare it specifically for this. And don't be intimidated by this PowerPoint, which was put together a few years ago for a talk that was an hour long. I will not take an hour. So I am a biologist. I study the evolution of development. So I, have done mainly worked with embryos, and I'm very interested in the evolution of embryos. And but I have some programming background before, and I always was interested in combining the two and doing simulations in biology. I dabbled in that earlier. And how to go fast.
Don't go too fast.
Don't go too fast.
Yeah.
Okay. Let me just walk through this and remind me of things to say. So that's me, and that's where I did this project in Austria at the Konrad-Lorenz Institute, where they were looking for people who wanted to do theoretical studies in evolutionary developmental biology, or what we call affectionately Evo-Devo. So I had been doing all the embryo stuff and had this idea for a project, and so they funded me, and I went over there and I did it. So in the big talk, I gave some introduction as to the biology I did and how I was taking two. I've done the two approaches. It's a nice little starfish juvenile. That thing's actually about 1 millimeter in diameter, which I grew up in a dish from fertilized egg. So I worked with starfish and sea urchins, and before that frogs. And whereas this is the new project, the computer project. So I am interested in the evolution of phenotypic novelty. Phenotype is your characteristics. All this stuff that he didn't talk about because he's talking about the genes, but the genes are interesting because of what they make, and so I'm interested in what they make. And these sea urchins I studied, and some typical sea urchin larvae, and some very atypical sea urchin larvae which develop in a different way, and that was the kind of work that I did before. And okay, this is just more of the same. I'll not get into that. But the evolution of body plan is a big topic in Evo-Devo. The body plan is sort of the overall structure of an organism. The typical body plan for us is being bilaterally symmetrical, having 2 arms and 2 legs, having a spinal cord, for example. Sort of major features, and different organisms have different ones. And the echinoderms, which includes that starfish there, and the sea urchins are very interesting because they're not bilaterally symmetrical. They're radially symmetrical, and the big issue is how did that evolve? They actually evolved from bilaterally symmetrical organisms, and in fact, if I go back to those larvae, their larvae, which is what a young one of these looks like, is bilaterally symmetrical, but it develops into something radially symmetrical. So, yeah, big topic. So, so I, through the years working on this, I was thinking I really want to get the computer modeling back to working on this, and how would you simulate the evolution of something like this? I tried to imagine like simulating a starfish. Well, that's really hard, as you were talking about. Or even something much simpler than a starfish. How would you simulate a whole organism? There have been simulations done in biology which are kind of different from the A-life simulations you're used to, but more theoretical biology approach, where you're simulating a lot of things about cell behavior, a lot of physics and mechanics of how cells interact. But they're always very specific about a particular cell type of a particular tissue. It's too much computation to try to simulate a whole organism. And I had the idea that it would be better to try to simulate something extremely simple and to actually evolve it from single-celled organisms. Now that sounds so cool that I better give the disclaimer that I haven't actually done that. But I wanted to get a start at what kind of model cells would you need in order to do something like that. So changes, let's see here, so evolution of body plan is just an example. There's the arthropods, by the way, that were mentioned earlier, not just insects, but also crustaceans. There's the echinoderm body plan that I worked on. That's the internal skeleton of a sea urchin. You've probably seen some of those. So, all right, let's just go ahead. Okay, so what can we do in silico? So it's Evo Devo, and my idea was if I could manage to simulate an organism that has somehow a life cycle and some kind of development, and then subject it to the typical artificial life approach that you're used to, construct a population of these individuals in a simulated environment, subject them to mutation and selection, and use this to explore evolution. That's kind of my goal. So I'm actually only working on step one. And so even though my goal is to do Evo Devo in the computer, this at the moment has no Evo in it and no Devo in it, because I'm starting small. So how do you simulate individual organisms? Well, the real important thing is the cells. And that's something that's often left out of a lot of the artificial life work that I've seen, because cells are complicated and messy. And sometimes this is somebody else's work. So a lot of you are probably familiar with it. This was a nice little simulation of getting complexity out of cells, simulated cells, but they were modeled as perfect circles. And you see a lot of this kind of thing. And perfectly circular cells aren't going to produce anything that's biologically meaningful. Because cells are not perfectly circular, and they're also not perfectly uniform, and they have specialized parts that interact, and those things are very important in the development of an embryo into an organism. This is from a project I did quite some time back that I alluded to earlier. When I first started graduate school, actually with the intent of doing simulations, I got to do some. And this is just to show the kind of theoretical biology approach that has been used, where so each of these is a different run, using different parameters over time. And when I joined the project, someone had generated this, and I experimented with what rules the cells would be following to try to get them to behave more like what was supposed to be happening. This is supposed to be the development of, in a frog embryo, something called the notochord, if you know what that is. It's a tissue that arises early in the embryo, and one of the things this does is it gets longer and narrower because of cell rearrangement. And we were trying to duplicate that.
Tubes.
And it doesn't make a tube, but anyway, it makes a cylinder though. This is what they had, and they didn't look anything like it, and by manipulating the rules that the cells follow, that doesn't look anything like it either, but this does look quite a lot like it. So that was successful. And this is just showing a blow-up of an individual cell, and that we were actually using a model of the forces going on as these cells interact with each other, where the arrows pointing outwards are hydrostatic pressure, and these arrows around the outside are tension in the membrane on the outside of the cell. So it's that kind of physics. So that's a great thing. But as I said, that kind of thing would be really hard to scale up into an evolving whole organism. We could barely do a notochord, let alone a whole frog embryo. And so I was looking for a simpler system where I could get some of that flavor, but combine it with the artificial life approach and get something that's evolvable. And right now, you talked earlier in the first, you talked a lot about vision, having vision. So I've got a vision for that could happen. I looked at a lot of different models that are used in theoretical biology for actual cell interactions, and I came up with this one, which I took a gamble would be the best starting point for me. And actually, before what should I say about that? The rest of what I have is pretty, it's down dealing with mechanics of cell interaction and cell movement. It's almost more fun to think about what I'm going to do with it when it works. I'm just trying to lay the groundwork for the basic physics and be able to start to add genomes and mutations and that kind of thing. And I'm also not a physicist. This was actually the hard for me, but I wanted to. This is the fundamental groundwork. So this model that I picked is somebody else developed this model. So I'm going to show you basically how it works, and if I have time, maybe show you some of the things I did to improve it to make it more towards what I'm looking for. It is based on a cellular automaton-like system. So, and in this case, I'm using a hexagonal array, but it's basically a bunch of elements. It's not exactly a cellular automaton because there are some pieces of information which are not stored in the array. So there's excess. But basically, it's a simple array. Every element has an index, a number in it, and a cell is simply defined as a bunch of adjacent elements that all have the same numeral in them. So all those ones, that's cell number 1, all those 2s are cell number 2. There's a certain kind of behavior. Cell 0 is the environment, which has slightly different rules than the other ones. And we can do this in color coding instead and make it easier to look at. So basically, this was put together by a physicist, and the idea it actually came from a model that had been used for other things in physics, but he made it better for doing biological cells. And the main thing is to work with the adhesion between cells, because cells are sticky, and that creates mechanical forces, and also just basic mechanical structure. So this is kind of how it goes. Repeated evolution, not in the grand sense, but evolution of this of this world, of this model. So your basic simulation goes like this. You select a point, and so a point and its neighbor at random from somewhere in the field, with a black arrow there. And it's an arrow because once you've selected those two neighboring points, you can copy one into the other. So step 3 is to do that copy, and so the red got copied into the white, and now the cells change shape. Oops, that's another thing. So what's step 2? You could randomly pick points and change the shape of these cells, and it wouldn't be very meaningful. Step 2, that just showed you another one up here, another example. Step 2 is to, after you've come up with these random points, to accept them or reject them based on energy minimization criterion. So now this is where the physics comes in, and the idea is certain things are likely to happen and certain things are not likely to happen, and it depends on the energy involved in the changes. So how's that defined? And I'm not going to take you through all the details of the math, but the energy has 3 components: adhesion, volume, and surface area. So the idea behind adhesion is that sticky things stick to each other, and that pulling them apart from each other is hard. So something that sticky things happen to come into contact with each other, they stay stuck. So that's a favorable configuration, is for sticky things to stay stuck. And the volume and surface area is the basic mechanical properties of the cell. So for one thing, the cell over a short period of time is going to have constant volume because it's basically water, and water is incompressible. It's also going to have constant surface area because the surface area is made of membrane, and it's finite amounts of that, and it's also under tension, so you can't just stretch it infinite amounts. So these are the constraints. Okay, thanks. Those are the constraints on the cells, and each time you do one of these moves, you can calculate based on having a certain amount of adhesion, what's the change of energy, and you've changed the volume and surface area, and so we're going to also create basically a force that maintains the volume and surface area. It basically gives it a springiness or a resistance to being stretched or compressed. And then these little lambdas in here are just, you could weight which ones of these are more important. So if these are more important, then they become very stiff and resistant to the stretch and compression. So not much elasticity. And whereas if you lower these values, then the cells become more elastic. And we won't go through that. This is something that's added to this. Somebody talked about stirring the pot. This is how I stir the pot. The obvious thing is if something's favorable, you do it, and if something's unfavorable, you reject it, because only favorable things happen. But the idea here is that things are happening somewhat randomly, and you could think of it as thermal energy, making some things giving a boost to energy here, and other things are lower energy. And there's an algorithm called the Atropos algorithm, which you can use to let things that are only a little bit unfavorable happen with some probability. So if this is a graph of the probability of occurring for different delta H, you'd start with the simple way is it's 1 over here and 0 over there. But instead, we have it trail off so that things that are only a little bit unfavorable have some probability of happening. And so that prevents, if you didn't have that, the thing would get into its local minimum of low energy, and then it would stop. And that would be boring. So, okay, so this is the simulation. This is actually a QuickTime movie of the simulation. And maybe I will show you first an actual piece of. Let's start with something simple. And as you see, I have those cells have membranes, but that was a later addition. So let me show you basically my implementation of what I found in the literature with only minor modifications. So there's a few cells, and that's fine. And basically, as you do this iteration over and over, they're just going to start wiggling around. The hexagons here are kind of small to see. And you can see they have kind of jagged edges because of those small hexagons. And each one of those is a cell started as a rectangle just because it's easy, but once you start it going, they have amorphous shapes, which is another thing that's hard to do. That's one of the reasons I liked this model, because it made it easy to do amorphous shapes. Now you notice that two of them have just touched each other, and they're sticky, and they're going to stay stuck together from now on. They touched each other only because the random movements happen to bring them close enough to each other. Once they're like that, they're like that. And if you let this run long enough, then eventually this one will come into contact with those. I mean, maybe sometimes it won't, maybe it'll drift off. But if it does, once it does, the ultimate lowest energy configuration is that they'll be arranged as a triangle with each cell touching each of the other two, and they'll never leave that once they get into it. So this was basically, let me try different built in. In order to sort out and create a more interesting non-random structure, they don't have to do something deliberate. If the cells are of differential stickiness and just the random motion around that gets them to sort of reassociate with each other and sample their environment, will cause all the stickiest ones to be stuck together in the middle and engulfed by the less sticky ones. And so this model, I'll basically show it running. It takes about an hour or so to just run it and get something useful out of it. So this is the program. I'll go back to the PowerPoint now, because you'll get to see the QuickTime movies, which are run much faster. So that's that one again, same idea. Here's that one, and pretty quickly the green is in the middle and the blue gets to the outside. And so that's the basic POTS model and the basic biological function that it performs, which is pretty good. So then we got to, let's see, this is the part I should probably keep short. I think I've given you a flavor, but this is actually what I started with. It was kind of fun to, okay, I read about the thing and I talked to the guy who implemented it and who devised it, and I made my own implementations and so on. The next was to try to make it into something useful that would work better ultimately, be more realistic, because this has some problems that make it not totally general. And what I'm really looking for is so that I'm not stuck with a simulation like that notochord simulation that I showed you, where all I can simulate is notochords. I want something that's really general, where cells can behave in all kinds of different environments that make sense for cells to be in. And this was designed for basically studying cells in culture on a dish, and interactions kind of like that differential adhesion kind of circumstance. And I'm thinking about embryos, and I'm thinking one of the things that cells need to do in embryos is that they're not just uniformly sticky. And if they were, it wouldn't get much more interesting than that sorting out. What they have is localized patches of stickiness that are controlled and regulated by all the kind of gene networks that we were talking about earlier. And it's not really fully understood how cells crawl and so on. But to have an interesting simulation, you need to have, the cells need to be able to regulate their stickiness. You can put some sticky proteins out there on the surface, or take them back in, or have different parts of the cell that have different properties. And so what I want to do ultimately is have, first of all, membranes, because all this happens on a membrane. And membranes are important for a lot of other reasons, and you saw that I have some cells that have membranes. But then still, the membrane shouldn't be uniformly sticky. I should be able to have adhesive elements on that membrane that can be deployed in different ways by the cell. So first obvious step, how about cells that aren't sticky at all, onto which that'll be the background for having adhesive elements. And so finally you'll have a cell where patches are sticky and in between not. Because basically membrane itself is not sticky. It's the protein floating around in the membranes that are sticky. So I just made some adhesion equals 0 cells just to see what would happen. And so that's why they're gray. This is still the original model without my having done anything to it. And what would you expect?
That's weird. They turn to circle migrate on away from each other.
Well, they will not stay stuck to each other. They shouldn't.
Sticky, so
they should just disperse.
Yeah.
Exactly what I expected. And I didn't expect to have any work to do about this, but they didn't disperse. And it turns out there's an artifact. At first I thought it was a bug in my program. I must be doing something wrong. But it turned out not to be.
Bouncing off each other?
They're what?
Bouncing off each other?
Bouncing? I'm not sure what you mean by bouncing. But they're staying stuck to each other, basically.
It's because they're minimizing surface area?
No, it turns out it's because of the basic nature of that POTS move that I showed you, that underlying copy. If you think about it, and I should cut this short, but if you think about it, when you do this copy, it changed the location of the membrane on both cells. And so it's a coupled movement. Both cells are moving together. And so the only time they could come apart is if you happen to do a POTS move where the environment cell, which can also be involved, is copied in there. You can kind of peel it apart. But a lot of the moves are both cells, membranes moving together, and they're coupled. And so it naturally creates this
resonance.
This part.
It's a resonance.
Resonance, like you could say that. I call it suction.
Okay.
Inappropriate suction, not caused by real physical forces. So,
but you would need energy to separate, right? Or
it shouldn't be that if they're not sticky.
Right.
So basically, I came up with an algorithm that said, well, the other thing that you could happen if the red cell were pulling away from the blue cell.
Yeah, but I would expect if it's like random walk, it should be separate.
Exactly. Exactly.
So it's random walk.
Well, but it's not random in that the behavior of one cell is not random with respect to the behavior of the other cell.
Okay. So
Right?
there's no,
That's the problem.
There's no relation between them. No.
There's too much
The
relation.
blue doesn't know about the red, and the red doesn't know about the blue.
It shouldn't. Right.
So it can overlap.
Right. So what I did to solve that was say, well, if the red is pulling away from the blue, they might move together if they're stuck together, or they might move together if the blue cell is under compression and naturally wants to expand to fill into the space. It's another way they can move together. But if those things don't happen, then it should be able to just pull away and fluid fill in. So I say, let's say there's some of this fluid environment nearby, and an alternative move would be for it to be copied over that instead, like so. So then I evaluate two moves, and I evaluate the delta H, the energy change for both those moves, and I say which one's more favorable, and take that move. And just doing that one little change has changed everything. So then we have it behaving exactly like you'd expect.
Is this indicative of the search for water and the way water gives you that extra property to support life? Like we were talking about, we need carbon, but we're always searching for water, saying how does water impact what cells are doing and why is it so important? And does it...
I want to say no.
Throw it out there.
Be a wet blanket, so to speak, and say no. Because really that was just an artifact of the model. It was not really reflecting anything physical. I mean, this is just, water's not doing anything special here.
So did you do the equivalent of making an energy neutral interaction rather than an interaction which...
Well, that's what I hope to have done by making them non-sticky. Those are the only three forces in here are the non-stickiness, or the stickiness rather, the compressibility or incompressibility of the cells, and the stretchability or in-stretchability of the membranes. Those are the only forces. Oh, there are more things that need to be added. That's all that's in there.
So is there an equal probability for the move to move onto an occupied cell, like a part of another cell versus moving into empty space?
Equal probability. A point is selected at random, which could easily be a point outside a cell, and then one of its neighbors is selected at random.
Okay. And have you tried giving it a higher probability to move into empty space versus...
No.
So I wonder if that might...
We're thinking about.
Now, what's the state of this research now? Where are you?
Well, the state of this research. So actually, I worked on this a couple of years ago, and I've gotten a little bit sidetracked. Thank you for the lead into the plug. I got a little bit sidetracked. I've been doing some teaching, like heavy-duty adjunct, full-time, but paid like part-time teaching at a local college over in Moraga, if you know St. Mary's College. And I don't want to do that anymore. I want to get back to the research, and so you know, as an adjunct, I'm always looking for a job anyway. And now I'm broadening that out to look at non-academic opportunities, possibly. I want to find out what's out there. Academic would be good too, but my research is so oddball. I haven't found a home. So actually, I would really love to talk with any or all of you who are interested in this stuff, or one-on-one, just talk about what you do and the world you do it in, and where the opportunities might be. Yeah, that's the state of it. It
You're
hasn't
based
done
here
it.
then? You're
I'm based here, yeah. I mean, basically, the job in Austria where I did this was the perfect job, except for one thing: an absolute 2-year limit, and after that, couldn't be renewed.
Mm-hmm.
So I came back here, and that was a postdoc. So I've been looking for a faculty job, and it hasn't worked out that well.
The
But
AI
I'm here.
community, I mean, there are places where people who are doing artificial life of various sorts have faculty positions, right? There are places that are sympathetic to that.
I guess there are, and part of my problem might be not knowing where to look, because I know where to look for biology jobs, and I don't think they're mostly in biology departments. But I've looked at computer science too. I've seen a few that were. We should talk about this one-on-one, not do this as on the internet. But anyway, there were a bunch of other stuff in there.